So it's really a great honor to be part of this lecture series, both because of my interest in the social brain, also because, of course, the Simons Foundation has been so instrumental in really shaping the landscape, I would say, of neuroscience, but in science more generally. And actually, a lot of the work I'm going to present today was supported both by the Simons Foundation Collaboration for the Global Brain and for actually some of the trainees, the fellows who participate in this work, and I also even serve students. Okay, so one of the major interests of my lab has really been to understand the incredible diversity of behaviors that exist across the animal kingdom, both within and across different species. And so we've been interested to think about how neural circuits can adapt over different timescales to give rise to this incredible variation. On the one hand, circuits can be adapted very rapidly, even on a moment-by-moment timescale, to allow every individual within a species to appropriately respond to their unique and changing experience of the world. But of course, circuits can be adapted over much longer time frames, over evolutionary timescales, to give rise to fantastic diversity that exists across different species in the animal kingdom. And so what I'll tell you about today is how we've been really interested in thinking about whether there are really common principles in how circuits can adapt over these different time frames that would tell us something about the basic substrate for flexibility within the nervous system. And for this, I'll turn to social behavior and the social brain, because I would argue that there's no place where behavioral flexibility is more apparent or more relevant than in navigating the complex social landscapes that many species inhabit. From the fermenting fruits of Costa Rica to our own natural habitat, the fields of Central Park, as individuals, we're often challenged with, in these kind of complex social and dynamic environments, of defining with whom to interact and how. And I would say that this fundamental challenge is something that I think we can all intuitively appreciate if we simply reflect on how any individual conversation that we have, for example, in the picnic in Central Park, really reflects a tightly coordinated feedback loop, a reciprocal exchange of information between two interacting partners, where we must continuously adapt and modulate our behavioral response as we integrate and interpret the words of a partner and decide accordingly how to react. And so this kind of reciprocal exchange of information, of course, is very different if you are talking to a friend, somebody you know well, or if a colleague from work walks past and you realize that the social dynamic has to shift and the conversation takes a different form. And so as humans, I think this capacity for nuanced communication really is thought to have driven some of the evolution of some of the most sophisticated cognitive abilities. For example, our ability to predict and anticipate the reactions of another person, or our ability to actually infer and empathize with their experience and perspective. But of course, these reciprocal feedback loops are really inherent to all socially interacting creatures. And what that means is that we can take advantage of much simpler model systems to begin to understand really how social dynamics unfold through the interplay of evolutionary hardwired social arousal states and external sensory signals that animals are constantly experiencing that shape their social interactions. And so the behaviors that we've been looking at in the lab are the elaborate courtship rituals that male fruit flies, Drosophila, display in order to entice their females to copulate. So here you can see is a male fly that is courting a female. He orients towards the female, taps at her abdomen with his foreleg to sample pheromonal cues, chemical cues that cover her body. And if this discrete gustatory assessment indicates that she's an appropriate mate, as it obviously has in this case, he'll persistently pursue and sing to the female by extending a single wing and vibrating it to generate a species-specific courtship song that serves as a potent aphrodisiac for the female, who will eventually slow down and allow the male to copulate. Okay, so one of the really beautiful aspects of this behavior, of this duet, is just like a human conversation, that it really reflects a tightly intertwined feedback loop between male and female behavior. And that is to say that male behavior is really patterned under the influence of female sensory and behavioral feedback on a moment-to-moment time scale. And I think a really beautiful example of this is the fact that actually if you just simply plot the distance between the male and the female over time, you can see that initially the male is pretty much indifferent to the presence of a female until a discrete moment in time when he identifies her as a potential mate, and then he will persistently and very faithfully pursue her, keeping her at very close distance. And he does this because he's continuously modulating his velocity in turn step with her velocity. So male and female behavior are tightly correlated at these really moment-to-moment timescales, but they're also actually intertwined within feedback loops over much longer time frames, over evolutionary timescales through sexual selection, where, of course, female choice is thought to reinforce the emergence of male behavioral traits. And indeed, across the nearly 2,000 members of the Drosophila genus, there's been really rapid diversification of the male courtship rituals at almost every level to generate fantastic behavioral diversity, from the sensory cues that males use to identify an appropriate female mate to the motor displays that they perform in order to entice her to copulate. And I think a really beautiful example of this is that Drosophila suzukii is actually a close cousin to Drosophila melanogaster, where males have wings that are actually adorned with pigmented spots. And therefore, rather than pursue the female from behind, as you can see in Drosophila melanogaster, singing this courtship song, suzukii males orient towards the front of the female and perform this sort of silent visual display. Okay, so Drosophila is really powerful for the rapid diversification of courtship behaviors that they display, but actually even more important is the fact that we can gain genetic access to underlying neural circuitry. And that is because actually nearly all aspects of male courtship behavior are mediated by only about 2,000 neurons that are marked by a sexually dimorphic transcription factor called fruitless, providing really exceptional genetic traction to tracing these circuits from sensory input all the way through to motor output. Moreover, over the past few years, we've taken advantage of genome editing approaches to be able to translate the rich neurogenetic toolkit from Drosophila melanogaster to its close cousins, allowing us really to begin to understand how these kind of complex courtship rituals have been tailored to species-specific needs. Okay, so today what I'm going to tell you really is then kind of a two-part story. First, what I'll tell you about is a basic understanding of the logic that governs male courtship behavior in Drosophila melanogaster, really using the exquisite genetic tools that we have available in this one species to provide a basic blueprint for how mate choice is mediated. I'll then tell you very briefly about some of the work that we've been carrying out to do cross-species comparison and begin to think about how this logic has been adapted across closely related species. And finally, I'll tell you about some recent approaches that we've been taking to think about how courtship dynamics, social dynamics, are really altered in the context of the more complex social environments where Drosophila really meet and mate in the wild. Okay, so one of the important populations of neurons in thinking about mate recognition that I want to highlight is a population of male-specific neurons. There are 20 neurons in the brain called P1 neurons. These are obviously sexually dimorphic. They serve as a central node within the courtship circuitry that actually integrates information, multisensory information, and regulates courtship behavior. So they can actually integrate information from different kind of sensory cues that are emanating from another fly to control the male's sexual arousal and decide whether he's willing to court. And we were really initially very interested in thinking about these neurons in the context of their ability to recognize the chemical cues that indicate an appropriate sexual partner, the pheromones. And to do this, we recorded from the P1 neurons in a male fly, as you can see here, that is walking on an air-supported treadmill, a ball, like a little fly-sized treadmill. And then we can bring in the female abdomen on a tether and allow him to sample and taste the pheromonal cues that cover her abdomen. And you can see that every time the male fly taps and tastes the abdomen of his conspecific female, there's strong activation of the P1 neuron population. But far weaker responses or even inhibitory responses when he tastes the pheromonal cues that cover the abdomens of females from other species. So taking advantage of the really concise nature of these circuits, of the pheromonal circuits, and the fact that they're all labeled, as I mentioned, by this sexually dimorphic transcription factor called fruitless, we could trace actually the circuitry from the sensory neurons in the foreleg that detect these pheromonal neuron cues up to the P1 population. And we find that pheromone signals are actually routed through a kind of bifurcated circuit architecture where there's both an excitatory branch that can activate the P1 population and an inhibitory branch. And we think it's this balance of excitation and inhibition that really controls the chemical tuning, the pheromonal tuning of these neurons to guide males to court an appropriate mate, such that when a melanogaster male tastes the pheromones on his conspecific melanogaster female, it drives net excitation to promote courtship behavior. But when he tastes the pheromonal cues on a heterospecific female, a female of the wrong species, it drives net inhibition. Okay, so P1 neurons are really tuned to the pheromone cues that indicate an appropriate mate. As I mentioned, they drive courtship behavior. And this has been actually described in a number of different experimental approaches. The approach that we took here is to take advantage of really all the beautiful, exquisite genetic tools that are available in Drosophila melanogaster to express a light-activated ion channel specifically in this population and provide the male fly with a slowly rotating magnet. You can see the male fly is completely indifferent to the magnet until the moment we briefly activate these neurons using light. So this is a very brief optogenetic activation, but you can see that it totally transforms the magnet. The male is extremely attracted to it and will perform most components of the courtship ritual. You can see that he's pursuing it and even singing to it, despite the fact that obviously there's no appropriate behavioral feedback. The magnet does not reciprocate at all. Okay, so what this indicates to us is that P1 neurons, we think, drive a really enduring state of sexual arousal, where even a transient activation of this population leads to a lasting behavioral response and transforms the salience of visual cues to change the magnet and even another fly target from an indifferent object into a target of desire. And so we're really interested to ask, how is it that a male's arousal state controls the salience of visual cues? And to do this, to really begin to address this question, a great graduate student in the lab, Tom Hindmarsh-Sten, developed a kind of virtual reality system in which we could examine courtship behavior in tethered flies under the microscope. And to do this, he actually presented a fictive female to the male fly that was projected onto a visual panorama and rotates back and forth at a constant angular velocity. And I should point out that one of the really great advantages of this system is that in contrast to the complex interplay between males and females, this kind of tight feedback loops that occur in the context of natural courtship, in this context, the female is basically invariant. And so we can really naturally uncouple changes in a male's internal arousal state from changes within his sensory environment and really think about how the salience of visual cues can be altered by changes in a male's arousal state. Okay, so here you see a male fly that is watching this visual target sweep back and forth and back and forth. We briefly optogenetically activate the P1 population for just a few seconds, and you can see that the male begins to track. And he does so really with remarkable fidelity, going left and right every time the visual target sweeps across the male's visual field, all the while performing the unilateral wing extensions and vibrations that are a hallmark of the production of the courtship song. So what we find is that even with a very brief optogenetic activation, we activate these neurons artificially for just a few seconds. It can lead to actually many tens of minutes of courtship pursuit, as you can see from this raster plot here. Indeed, in some animals, they would track actually this visual target for almost as long as we presented it, up to 24 meters, which is as if you were to chase your partner for 17 kilometers, really reflecting the fact that this is a very motivated internal arousal state that we've created. But we can actually generate these kind of long-lasting arousal states, not just through these artificial mechanisms, but even by providing the male fly with a female on a tether to allow him to sample her pheromonal cues and replicate this initial kind of gustatory assessment that he carries out. And actually, quite remarkably, even just by changing the statistics of the visual stimulus to more closely replicate what the male fly would experience as he was chasing the female in a courtship assay. So now that there's a translational component as well. Male flies, we found, surprisingly, will begin to spontaneously court this visual target. And what this means to us is something quite striking, which it suggests actually that in the absence of any other sensory information, males can actually take just advantage of the visual target and become aroused and interpret as a female and begin this courtship pursuit. So one advantage of this is that we can now begin to actually record from neural populations within the male fly brain to think about actually how neural activity is transformed as males become aroused and begin to initiate their courtship. And so Tom recorded from the P1 population as males began spontaneous courtship pursuit. And what we found is that actually the activity of these neurons is really tightly aligned, not with the onset of the visual stimulus, but really with the onset of the male's initiation of courtship. In accord with the idea that once these neurons are activated, that they will drive this long-lasting state that drives males to pursue visual motion. But what was quite striking to us is that these neurons actually did not stay persistently active, but they fluctuated in a manner such that their activity was really tightly correlated with the vigor of a male's courtship pursuit on a moment-to-moment time scale, not with actually the speed of the target or the visual target, or the speed of the male or the visual target itself. And so this suggested to us actually that these neurons have a really interesting property that they kind of effectively signal at two different time scales, When activated, they initiate this long-lasting arousal state that drives courtship pursuit. But then on a moment-to-moment time scale, their activity is reflecting the moment-to-moment changes in a male's vigor and his kind of the fidelity of his pursuit of this virtual target. And so what I'll tell you now is that actually this is really interesting to us because what we find is that the P1 populations serve to really dynamically modulate the gain of a concise visual motor circuit to allow males basically to track female stimuli only when they're sexually aroused. Okay, so a key component of this circuit is a population of neurons termed LC10A neurons. These are visual projection neurons. They carry information from the optic lobe of the fly in a kind of retinotopically organized array, and they send this information into a discrete optic glomerulus within the brain. And previous work had been suggested that these neurons are really tuned to small spot visual motion, like the motion of a female target. And these neurons we find are essential for this kind of faithful pursuit of males, as you can see here, because if we optogenically inhibit these neurons, now silence them, as I'll show you now when we turn on the LED, these neurons are now silenced, and males basically become effectively blind to the presence of the female who will now just basically walk away. So LC10A neurons are really necessary for the kind of faithful visual pursuit, but what was surprising to us is that when we recorded from these neurons, their activity was very low to the visual target. They were not actually that visually responsive. That is, until the exact moment that males initiated courtship pursuit, and then the gain of these neurons was strongly amplified, such that actually their activity was very high every time the visual target swept across the male's visual field. And so if males actually transiently suppress their courtship or stop tracking, you can see that the gain of these neurons goes back down to baseline, so they're no longer responsive to the visual stimulus. But then if they reinitiate courtship pursuit, you can see that their gain is increased once again. And so on a moment-to-moment time scale, we think that the gain of LC10A neurons is strongly amplified by a male's arousal state, such that their activity is very high when males are courting, not when they're just simply running around. So very much a strong state-dependent modulation of this population. Okay, so to understand how this kind of change in the gain of these visual neurons may actually be propagated to the motor systems, I can say that we can take advantage of one of the great resources of Drosophila in modern neuroscience is the fact that we have a whole brain connectome, in fact, multiple whole brain connectomes, that allow us to trace the flow of information through really specified neural pathways with exquisite resolution. And so if you look in the connectome of the brain, you can actually find LC10A neurons, and you can look at all the populations that are downstream of it, where it sends information. And what's interesting is actually this population of visual projection neurons is only 2 synapses from an array of descending neurons that carry information from the brain down into the ventral nerve cord, which is like the spinal cord of the fly, and is poised to really provide signals to the musculature of the fly and control its steering behavior. And indeed, if we record from one of these neurons, DNAO2, which has been previously implicated in steering behavior in the fly, we can see that the activation of this population of descending neurons is basically perfectly replicates the activity patterns of the LC10A neurons, as if the population, these neurons, the LC10A neurons, are really faithfully transmitting this information down to this descending population to allow for moment-to-moment control of the steering behavior, such that these neurons are activated every time the male fly turns in the ipsilateral direction. So to understand whether this sort of very simple architecture and this sort of gain modulation mechanism could explain the visual pursuit of a male fly, Tom built a very simple kind of computational model where he structured the excitatory input of LC10A neurons using the published spatial temporal receptive fields, so the basic functional properties of this LC10A population, onto a simple kind of circuit architecture, a simple little network. And actually using this very simple model, it could really predict the moment-to-moment turning behavior of males when they were tethered and highly aroused to either relatively simple or more complex visual stimuli. But what was quite striking is it was even able to predict the moment-to-moment turning behavior of male flies in free courtship, where we would basically take the projection of a female the trajectory of a female projected onto the male's retina and use it as input to the model. Okay, so taken together, what this says to us is actually that this sensory motor circuit, the LC10A sensory motor circuit that I described, this very simple pathway, is basically deterministic. That is to say that we can really predict the behavior of the animal if we understand what the visual input is into the LC10A pathway. And what that might imply is that once males are aroused and this pathway is engaged, the gain of this pathway is enhanced, actually males will pursue almost any moving visual target. And so to give you an intuition for actually really how stereotyped these kind of social behaviors, these courtship behaviors can be, once we get rid of female variability, we took advantage of a system that we recently developed, which is a two-dimensional projector system, where we can basically project any arbitrary visual stimulus onto the floor of an assay chamber. And one of the advantages of this system is that we can take real female trajectories, like the one I show you here, and we can project it onto the floor of the chamber, allowing males all males to have access to the same visual stimulation with taking out the female variability. And I should say that this was the work of a great graduate student, Florian Hollander, and actually a Simons fellow, Philip Brand. So here I'm showing you is a movie of 4 flies that are given the same visual target, and we then activate the P1 neurons to make these males all highly aroused. And you can see in this context, actually, courtship behaviors become almost deterministic, not only in how the male modulates its its velocity in response to changes in the velocity of this visual target, but even which wing they choose to sing with. And so this suggests to us really that actually once males are aroused with a very determinist or very invariant visual stimulus, their behavior becomes highly stereotyped, suggesting that the variability that we see in many cases really depends on fluctuations in a male's arousal state. And indeed, that is because, as I mentioned to you, this entire sensory motor circuit that is highly deterministic is under continuous modulation by the P1 population, which does not act really like a switch to turn males on, the arousal on, but actually serves more as a rheostat to control the gain and the kind of strength of the way visual information is being propagated through this pathway on a moment-to-moment time scale. And a really nice bit of evidence for this is that if we perform dual recordings of the P1 neurons and the LC10A neurons while males initiate spontaneous courtship pursuit, you can see that their activity is really highly correlated on a moment-to-moment time scale. That is to say that the activity, the magnitude of the LC10A response as the visual target sweeps across the male's visual field is directly proportional and predicted by that P1 activity at that very same moment. Okay, so P1 neurons, we think, therefore serve to have this kind of dynamic gain modulation of these visual motor pathways. And I'd say that P1 neurons as a site of multisensory integration offer a very powerful nexus for such dynamic control because they can provide additional information about ongoing sensory and behavioral feedback to control when males will track and what they will track. And to give you really an intuition for the importance of having this ongoing feedback in the system at all moments, I'll show you what I think is quite a fun experiment in which we now provide the male fly in this projector system, a wild-type male, this is just a normal male fly, with 2 visual targets that are moving on the floor. And you can see that the fly basically is totally indifferent to the presence of these 2 visual targets. I think, in fact, he might even be a little bit freaked out by them as they pass over him. But then we give the male fly a real female, a conspecific female that has the pheromones that excite the P1 neurons and activate and make him become aroused. And you can see that the male fly in this aroused state not only courts the conspecific female, but he actually often courts the visual targets that are on the floor. So even in trying to discriminate between a 3-dimensional female and a 2-dimensional target, males are actually have a difficult time once they're aroused, really highlighting actually that they're very sensitive to visual motion. Okay, so P1 neurons are this interesting kind of site of multisensory integration that then have the capacity to control the gain of visual motor pathways and prevent this inappropriate courtship pursuit. And in that concept, we were particularly interested in the role that heterospecific pheromones play in regulating the P1 activity. Because as I described to you, I already showed you that there are inhibitory pathways that can suppress the activity of P1 neurons that directly impinge, these inhibitory pathways directly impinge on the P1 population. But most importantly, pheromones across the Drosophila genus, the female pheromones, have really rapidly and repeatedly diversified as signatures of species identity. And that means that basically every female carries a slightly different pheromone that can serve it to both provide excitatory cues to promote courtship behavior, but even more importantly, inhibitory cues that prevent this inappropriate courtship pursuit. And so the inhibitory pheromones, we think that if they inhibit the P1 neurons, they should reverse the gain of this visual motor pathway by suppressing kind of how strongly this pathway can drive the activity of descending neurons. And indeed, some bit of evidence for this is the fact that, or at least one example that kind of highlights that idea, is that why I showed you that males cannot discriminate very effectively once they're aroused between a conspecific female and a two-dimensional target. They do a really good job when presented with a conspecific female and a heterospecific female that carries these inhibitory pheromones. So you can see this male is in the process of courting its conspecific. It encounters a heterospecific female, but it quickly rejects her. We think because he becomes transiently blind to the presence of this heterospecific female, reorients and finds a new female. And indeed, if we make flies that are basically mutant for all pheromone receptors, so they can't detect either conspecific or heterospecific pheromones, we find that these male flies that are mutant for pheromone receptors that can't detect these inhibitory pheromones, these males are totally promiscuous. They are equally attracted to both conspecific females and heterospecific females, who are all equally attractive to them and drive their robust pursuit. Okay, so what this suggests to us, really then taken together, is that the logic of male mate choice in Drosophila actually goes a little against our counter to our intuition, where we might have expected that conspecific females carry a set of really unique instructive cues that signify that she's the appropriate mate and allow males to target their pursuit towards these conspecific females. But instead, what we find actually is that basically all females are inherently attractive visual targets, and male pursuit is then honed by pheromonal inhibition. So it's almost by subtraction that males are able to keep track of the appropriate female targets. So this kind of interplay between then general visual arousal and very specific pheromonal inhibition is what allows males to basically maintain pursuit and sample across different females and identify the appropriate female mate. And we think that this interplay is actually particularly important in the context of the natural environment, the more complex social landscapes in which flies actually normally meet and mate. And I should say that in general, we become increasingly interested in the lab in thinking about the kind of sensory constraints that flies or all animals encounter in the context of their natural environments. And something that we become particularly inspired by really actually going to take field videos or studies of flies in the wild. And so Philip Brand, Anna Reba, and Julie Ree, 3 members of the lab, went down to a field station called the Field Station La Gamba in Costa Rica, which was previously described as Droso Rica, due to the great congregation of different Drosophila species that all are endemic to this area. And they took a number of really kind of inspiring field videos that helped us to better understand the complexity of the natural landscapes in which flies meet and mate. So this is a very typical video of a rotting plantain, and you can see just what a complex visual environment flies have to contend with. That an aroused male fly, his first kind of instinct is to doggedly pursue almost any kind of visual moving object that comes his way. And then he goes over to taste the pheromones that cover these different targets and decide whether she's an appropriate mate and worth continuing pursuing, or will abort his pursuit and actually reorient to sample the pheromonal signals on another female. Okay, so what I've described then really is how we think there's really a kind of a segregated circuit logic in Drosophila that underlies mate recognition that really reconciles 2 competing needs for mate pursuit and discrimination. So first, what I described is that there's a very almost deterministic, high-fidelity visual motor pathway that assures male flies, once aroused, can be extremely faithful in their pursuit of a conspecific female, to pursue her basically until consummation, to render her sexually receptive and ensure he has access to a mating opportunity. But this entire visual motor pathway is under continuous gain modulation by a population of P1 neurons that provide ongoing sensory behavioral feedback to make sure that their pursuit is targeted towards appropriate mates. And in particular, what I showed you is that heterospecific pheromones carried by females of other species, by providing inhibitory input into the P1 population, we think serve a very important role in assuring males hone their arousal, hone their pursuit towards appropriate targets, and suppress pursuit of inappropriate female mates. Okay, so this is the logic that we started to elucidate in Drosophila melanogaster, but we can now ask, how has this logic been adapted across the different Drosophila members of the Drosophila genus? And I just want to remind you that while there's really fantastic behavioral diversity in the kind of courtship displays that males perform across these many different species, for example, between the very kind of subtle motions of Drosophila mauritiana to the really acrobatic and energetic approaches of Drosophila elegans, that despite this incredible behavioral diversity in these motor displays, actually males of all these species fundamentally have to perform the same behavioral program that is essential to their reproductive success. That is to say that they have to become motivated, identify an appropriate female target, become motivated to pursue her until consummation. And so we were really interested to think then in this context of how actually the sort of logic has been adapted that we described for Drosophila melanogaster, has it been adapted across closely related species? And in particular, what is the role that P1 neurons might play across these different species? So Rory Coleman, a postdoc in the lab, translated the same genetic drivers that label these P1 neurons in Drosophila melanogaster into the genomes of these close sister species of Drosophila melanogaster to label really what we think are homologous neural populations. And though they anatomically look a little bit different, one of the really great aspects of having genetic access to these populations is that we can now test their behavioral role. And what he found is that optogenetic activation, this brief activation, exogenous activation of this population in all these different species drove the same persistent courtship pursuit of inanimate targets, these either magnets or projected dots. So taken together, what this says is that actually we think that P1 neurons play a really conserved role in promoting sexual arousal across species, raising really interesting questions about how diversification of the pheromone pathways has occurred to allow males to hone their pursuit towards appropriate conspecific females, and that not every fly of different species was always going to be pursuing the same sort of visual targets. And in that context, it's really important to just remind you that what I described already is that actually female pheromones have rapidly and very rapidly and repeatedly diversified across the members of this genus. This includes, and this rapid diversification actually gives rise to both kind of novel conspecific pheromones carried by females of their own species that excite the P1 neurons to promote a male sexual arousal and initiate this process. But even more importantly, I would say, is the emergence of unique heterospecific pheromones that inhibit the P1 population and basically reverse the gain of this visual motor pathway to prevent inappropriate courtship pursuit. And so while I showed you that P1 neurons across these different species, across these 4 related species, are all sufficient, they play a common behavioral role in driving a male to become sexually aroused. Actually, when we look at the pheromonal tuning of these P1 neurons across these different species using functional calcium imaging, what we find is that they all display distinct and species-specific pheromonal tuning. That is, the P1 neurons are really tuned specifically in a highly species-specific manner to identify what is an appropriate female mate. And from taking advantage of the concise nature of these pheromone circuits, and the fact is, again, as I told you, that they're all labeled by this one sexually dimorphic transcription factor, we were able to start to really do comparative analysis of these pheromonal processing pathways across these different species. And what we find is that the architecture of pheromone circuits is really highly conserved. It basically is maintained, we think, across at least simulants melanogaster and Drosophila yocuba, where pheromone signals are routed through both an excitatory and inhibitory branch. And interestingly, we think that the differences in pheromonal tuning that we see across these different species not only reflects differences in the way these pheromones are detected at the level of receptors, but more importantly and really interestingly, that it's related to actually how there's been subtle retinkering or tinkering with the kind of balance of excitation and inhibition at the level of the P1 population, such that conspecific pheromones that promote excitation in one species can actually be interpreted in males of another species as heterospecific pheromones by activating this inhibitory pathway more strongly. So through this sort of balance of excitation and inhibition, the reweighting, kind of subtle reweighting and subtle changes of a common and conserved circuit architecture, you can actually get species-specific interpretation of pheromonal cues. Okay, so what I described then is that P1 neurons, we think, actually serve as a very interesting substrate for the rapid evolutionary diversification of mate preferences across these different species. They play a conserved role in controlling a male's sexual arousal, but it actually intuitively makes sense that by just changing the weighting of different sensory pathways onto the P1 population, it allows for the rapid emergence of kind of species-specific mate preferences. And so we think that actually it's quite interesting to consider that there are going to be potentially some kind of favored targets for behavioral evolution, like these integrative nodes, that can allow for the rapid diversification of behaviors by actually changing the way that they weight different sensory information to control internal arousal states and the patterning of behavior. But it also starts to say to us actually how this P1 population plays really a dual role, not only in shaping the evolutionary kind of preferences of males across different species, but also, as I described before, in the moment-to-moment patterning, providing really kind of an idea that these sort of central integrative nodes really are a very key substrate for evolutionary and or behavioral flexibility over different time frames. Okay, so I described to you then that we started to gain a really good understanding, I would say, or an initial understanding of how male courtship behaviors are patterned by ongoing changes in a male's internal arousal state and changes to his sensory environment. And in the last a few minutes of the talk, I'd like to highlight that we started to think a little bit more about how females interpret the performance of these male, or assess the performance of these male courtship rituals to identify whether they're an optimal or an ideal mate. Because we think actually the evolution of these courtship rituals really was actually intended to satisfy choosy females and allow them to assess and evaluate the different males. So this idea that females might be evaluating and assessing between different males is something that actually became particularly important to think about in the context of the natural environment. Because as I showed you, for example, on the rotting plantain or in the natural kind of social landscapes for Drosophila, you can see that many males and females congregate on a single food patch. And what that implies is that females are rarely pursued in isolation, actually, but by multiple males synchronously, allowing actually females to assess and evaluate which is a better and more ideal mate. And so we found that the kind of complex dynamics that you can see in the natural environment, actually we could replicate a lot of them actually by just adding one more male to the mix. So here you see that have two males that are now Drosophila melanogaster males that are now competing for a single Drosophila melanogaster female. And I hope you can appreciate that the dynamics of courtship are really strikingly different. Everybody moves much faster. They all perform the same kind of basic behavioral components of courtship that we see in the sort of kind of tight feedback loops of male-female courtship. But we also observe the emergence of a completely novel kind of motor display that only emerges in the context of these competitive courtship environments where males perform these very rapid wing flicks that we think serve an important role as signifying an agonistic signal that actually can provide them with a brief competitive advantage. And so this provides us with the opportunity then in these competitive courtship examples to think about how do females then select between these competing mates? How does she choose between these two males to identify the optimal male? And so this is the work of Tom Heinmeier-Sten with another great graduate student, Rufei Li, who's defending her thesis in 2 weeks, along with Shadi Elizier, who is actually a surf student who worked very closely with Tom. And so what they started to think about is that certainly one of the key kind of components of courtship behavior that has long been known to be essential for female sexual receptivity is the courtship song that males perform, as I described, by vibrating a wing to generate this sort of sinusoidal humming that has very species-specific statistical properties. So the spectral properties of the courtship song are also very rapidly evolving and allow females to identify males based just on hearing these signals. And indeed, this courtship song is known to drive an auditory circuit that triggers females to open up her vaginal plates, which is the final motor element that is required for copulation to ensue. And this is under the control of a concise visual auditory circuit that has been beautifully described by work from Barry Dixon's lab, which contains a single descending neuron that directly triggers the vaginal plates to open. So an indication that the courtship song plays such a critical role in controlling female sexual receptivity comes from experiments like this, where we can look at how females copulate over time, or whether females are willing to copulate with males that are not able to sing. And in flies, we can do this very simply by surgically removing the wings of a male to make him basically mute so he can no longer sing. And you can see that actually females are very unlikely to actually copulate with a wingless male that can't sing to her. But interestingly, we can simply just play back the courtship song of a conspecific male and you completely restore her sexual receptivity. So this says to you that the courtship song that males perform is an essential and key component of the male's courtship ritual that she's attuned to control her sexual receptivity. But surprisingly, what we found is actually females cannot discern which male is serenading her within these competitive courtship environments. And to demonstrate this, we actually performed a very simple experiment where we took a female and offered her the opportunity to copulate with either a winged male who can sing a perfect rendition of the courtship song and a wingless male. And what we found is, surprisingly, the wingless male copulates with the female a really large fraction of the time. And he does so in a really interesting way, which is that he sneaks in right at the moment where the winged male sings to the female, which we in the lab decided was really the very definition of a wingman. So wingless males actually copulate with the female in these competitive assays a large fraction of the time. But if you consider contexts in which both males are very close to the female, where the winged male can actually serenade the female and allow the male to sneak in, it's basically 50-50. The female cannot discern which of the males is singing to her. And so this really highlighted and brings to mind, of course, that there are two components to sexual selection as conceptualized even since the time of Charles Darwin. Not only are there very key components of a male's kind of morphological or behavioral kind of pattern that are served to charm the female and promote her sexual receptivity, but equally important are actually components that actually serve to drive away or repel rival males. And just as this courtship song serves to promote sexual receptivity in the females, actually to another male it indicates the presence of a rival and promotes the male to perform these bilateral wing flicks, which serve as a steric kind of cue that repels other males away, and therefore serves as kind of an agonistic action that should provide the males with a transient advantage. So to further probe the role of these kind of bilateral wing flicks, we took advantage of the fact that could find a driver line, a genetic driver of the neurons that drive these flicks in males in the ventral nerve cord. That once you silence these neurons, males are not able to flick anymore at all, but it does not affect their singing in any form. And this highlights that there really are two distinct modes of acoustic communication that these males are performing. They sing towards the female in one mode of communication, but they also are flicking to other members of the triad through these bilateral wing flicks. And we think that this is really indeed a form of acoustic communication because actually we can optogenetically activate these flicker neurons. And you can see that actually when males flick, it produces a very distinct acoustic signal with completely different spectral properties compared to the courtship song. So as a consequence of the males flicking almost continuously in the context of these triads, in the context of competition, if you record the soundscape and compare it between a courting pair and a triad, you can see that actually it's a much more complex acoustic environment in this context of the triad. And this is largely due to the male's capacity to flick because you can silence the flicker neurons, and you can see it returns the sort of the kind of properties, the kind of statistical properties of this acoustic landscape. Really, it renders it back to the kind of simple dynamics of the courtship pair. And so this was interesting to us because it suggested that I told you that females are very attuned to the presence of the courtship song, and they're sensitive to the specific spectral features of a conspecific male. So it made us wonder whether or not actually, can they identify a courting male in the context of this complex acoustic landscape? And so to examine this, we looked at the circuits that underlie vaginal plate opening in females, which are comprised, as I told you, of a vaginal plate descending neuron that drives the motor program of the female to open her vaginal plate. And inputting onto this descending neuron is an excitatory population and an inhibitory population. And so if we record from these two excitatory and inhibitory populations and play back to a female the acoustic landscape from a male-female pair, so just basically the courtship song, or the more complex acoustic environment that is presented by the triad, by these competitive courtship landscapes, what we find is that, not surprisingly, when females hear the courtship song, it activates the excitatory branch much more strongly than the inhibitory branch to promote sexual receptivity. But when the females hear this more complex acoustic landscape, this more complex signal, it drives the inhibitory branch more strongly than the excitatory branch. And so what it suggests to us is that actually these bilateral wing flicks really serve, we think, as a signal to basically effectively jam the auditory pathways of female and prevent the female from recognizing and perceiving the courtship song of an appropriate rival male. And this is something that we also see at the behavioral level by looking at the vaginal plates of females in these two different social environments. So you can see that every time the male sings, there's often a lengthening of the abdomen of the female, which is correlated with her opening of the vaginal plate. But this is strongly attenuated in the context of these more complex social landscapes. So one thing you might be wondering then is, well, how in the end does actually a male ever succeed in mating, right? It seems almost like an impasse where actually both males are continuously singing and trying to gain an upper hand in the context of competition and preventing the female from hearing the appropriate courtship signal from their rivals. And so what we find is actually that really what is important in order for copulation to ensue is that in the context of this competition, males need to sequester the female just for a moment to actually overcome this acoustic interference and allow him to be the sole male to serenade the female, to trigger her vaginal plates to open and allow her to sing. And indeed, actually, this ability to gain a very brief upper hand from the rival is something that is really facilitated by the ability of males to perform these wing flicks. Because if we silence males, we present now a female with a wild-type male and a male where the flicker neurons are silent, so they can only sing, these males. They cannot flick. This male, you can see, is at a great disadvantage. And in fact, he loses a large fraction of the courtship trials. Okay, so in just taken together, what I want to highlight really is the very kind of important aspect of this kind of interplay that males are performing all the time in order for their reproductive success. That is, that not only do they have to continuously alternate between singing towards a female to promote her sexual receptivity and drive her vaginal plates to open, but they also must continuously actually perform these bilateral flicks to actually prevent their rival from gaining access to the female. And so we've been really interested to think about how that kind of behavioral flexibility, this kind of social flexibility, is really engineered into the nervous system of males. How do they alternate between these two different opponent motor programs on a moment-to-moment time scale? And so what I showed you before is that males will actually perceive a visual target as a potential female and courter at any moment. But what we find is that if they also hear the courtship song, their perspective of this visual target, of how they perceive it, is totally transformed. And so here you can see is a male fly that is actually singing to the visual target. The wings go back and forth. They perform the bilateral wing extension. But if we play back synthetic courtship song to that male, you can see that he charges forward and performs these bilateral wing flicks on a moment-to-moment time scale. And so every time we play the song back to the male, he increases his forward velocity like he's about to charge towards the fictive target as if it's a rival male, and performs these bilateral wing flicks. But then in between the moments when we're playing the pulse song, he goes right back to courting as if the female was just present. And so this suggests an amazing level of kind of behavioral control and flexibility that is intrinsic to these circuits. So what this suggests is that in the context of these complex and competitive social dynamics, visual motion does not simply get transformed into courtship behaviors, but can be rapidly rerouted into alternative motor circuits to either drive courtship or aggression, depending on the presence of additional sensory cues, for example, the male courtship song. And we think that these two pathways, the way that this system works sort of almost like a switchboard to route the same visual information into these different motor pathways, is really under the control of two distinct higher order nodes. I described one of them as the P1 neurons that become activated by actually the presence of a rival male to actually encourage him to maintain persistent arousal and persistent courtship pursuit. But then on the other branch, there's another population of neurons that triggers these wing flicks that actually can route this signal into an aggressive mode and allow the male to perform even transient flicks that get layered onto the courtship ritual to allow him to actually transiently try to repel his rival and gain an upper hand. And these neurons actually are just, as I said, transiently engaged in this pathway. Okay, so just to provide you with a few kind of final conclusions, what we've described then is really how if we start to think about actually kind of the social dynamics of even a simple model system like Drosophila, you can see that in the wild, much more complex interactions occur, including these kind of agonistic competitive interactions that you don't see with just in pairwise animals. And what we start to think about in this context is how really mate choice was something that we thought we could really begin to disentangle and understand, turns out to really reflect a kind of complex interplay between male competition and female choice. And in fact, in the context of these competitive triad environments, it's actually quite difficult to decide who has agency in these mate choice decisions, and probably it's distributed across both of them, males and females. And finally, females, although we don't think that they're able to probably distinguish between which male is serenading them, what I described is that in order to be reproductively or competitively successful, males must be very vigorous in their pursuit and continuous in their pursuit in order to be able to gain access to this one moment in time when they're able to gain reproductive access to, or optimal copulatory access to the female, suggesting that she might still be indirectly selecting for males that are the most vigorous in their and persistent in their courtship. And so finally, I just wanted to highlight that actually what we start to think about then is really the flexibility of social behaviors is a very core feature of these circuits. And just like you can modulate your behavior on this moment-to-moment time scale as you navigate through the Sheep's Meadow in Central Park, fruit flies, Drosophila, are able to do the same sorts of kind of moment-to-moment modulation by actually taking advantage of how the kind of ongoing changes in the external sensory signals and the interplay between that and their long-lasting arousal states. And so we think that these really end up being basic building blocks of how the social brain functions. And so with that, I'll just close and thank the members of my lab who carried out this work, who I think I named along the way. We've had great collaborators and input from a number of different labs. And I just want to thank the Simons Foundation again for their generous support of many of aspects of this work and for the opportunity to be here. So thank you so much.
Thank you for a wonderful talk. So you have time for some questions? So let me begin by asking about when you have two males, do they sing the same song?
So we have not, in general, we believe that they do sing the same song. It's interesting, actually, males do not actually overlap that often in their song. It's as if it's not that they're trying to sing over each other. We actually have not dissected out how much they can modulate their song in the presence of a rival if they are trying to change the acoustic features. Partially because I think these are actually really hardwired elements that the female is very much attuned to. But it's an interesting question that, other work from other labs, including Mala Murthy's lab, has suggested that there is this sort of capacity for modulation based on actually the sensory environment. And so it is an interesting idea that it could be that some statistical features or the loudness could be changed. So.
Just hit the button or maybe easiest to on that.
Okay, thank you.
Good.
Thank you for the interesting talk. I was also curious about male-male interaction. You mentioned that pheromones of a different species kind of turn off the pursuit of male. I was wondering if males of the same species also have some kind of chemical cues or pheromones that could turn off P1 neurons, in the same way.
That's a very good question, is sort of thinking about actually what kinds of cues males might be using beyond the song. Are there like more proximal cues that they could use, like pheromones? It turns out that actually we think all males of all species, or many species, produce the same volatile pheromone. It's a unique compound called cis-vacinyl acetate. And indeed, actually, we think that it is very important in modulating actually these circuits to actually promote sort of more aggressive states and suppress courtship states. So there is also a chemical component of that as well. Yeah.
Yeah, thank you. That was really interesting. So I guess I'm trying to understand, in the case where you just cut off the male's wings and they're successful 50% of the time, I mean, that suggests a strategy to be successful 50% of the time is to neither sing nor do bilateral wing flicks. And, maybe it's just, too complicated game theory for the flies to figure out how to optimize this, but I don't know if you have any thoughts on.
No, it's an interesting question to think about. I would say that one aspect of it that probably is worth considering is actually in the context of the natural environment. It's not just two males against one female. It's actually probably many, many males that are all pursuing the same female target. And in fact, in the wild, it's thought that they're very rarely virgin females on a food substrate, that most females are mated, which makes it even a much more challenging and highly competitive environment. So, for example, one thing we've been thought about is in the context of why they continuously go back and forth between these two to gain opportunities. And I think one thing to consider is the fact that, for example, if we never, for example, aggression in Drosophila has been actually studied for many years, many decades even, often in the context of competing over a food substrate, where actually two males escalate into much more vigorous aspects of aggression, like they lunge at each other. We never see that in the context of these competitive interactions, partially because I think both courtship and aggression are sort of held in check in order to, because if they were to spend too much time fighting each other, actually a third male would swoop in, or if they spend too much time courting, actually they get basically brushed away. So I think it's actually a very interesting question, and probably something that could actually take advantage of more kind of, I would say statistical modeling, and to think about optimal strategy. I would say that also in the context of where just two males, one very localized chamber, and one female, it may not represent the natural dynamics exactly either.
Great talk. Have you studied the topology of the P1 network, and if so, is it static?
Yeah, it's an interesting question. I mean, I think one of the kind of elusive aspects of the P1 population that we have not yet understood is, how does this population of neurons drive this long-lasting state when their activity is not actually reflective of a long-lasting state? So is it downstream, or is it in through some network recurrent circuit architecture? It's something that I think we're better poised to be able to do in the months to come, partially because what I described is we have a connectome. We have multiple connectomes, but they're all of the female brain, which is actually surprisingly informative about male behavior. But the P1 neurons, as in this male-specific node, are not present. And so thinking about the topology, their interactions, and how they actually may shape information, but also how they may feed back onto themselves is something that we're all, I think, many of us are very excited about with the emergence of the male connectome.
Hi, fascinating talk. So do we think if there's any determinants to the male success in this competition, say maybe things like the sperm storage, because I assume these males are promiscuous, so maybe lower sperm storage means lower chance at winning this competition, or maybe the other extreme, say the winning male always win.
Yeah, so there's interesting questions about what may control male success that is beyond just this moment-to-moment interplay. I'd say that there's actually a whole aspect of this that we have not totally examined, which is in the wild, again, we pitted two males that were basically isogenic, raised identically, so that they would be the best match, so we could think about behavioral performance. But if, for example, you take a male that has been socially isolated versus one that has had many sexual partners, the motivation of the male with many partners actually goes down. This is work from other labs, like Mike Crickmore's lab, that has described how they become less motivated to both be aggressive or to court. And so you can imagine that puts them at a disadvantage. So there are really interesting questions about the natural variation that exists in the wild and how that might allow actually for these sorts of kind of courtship competitions to have much more obvious outcomes. I will say that I don't know, for example, that we have any evidence that, like there are any kind of hierarchies that are formed, but we haven't really looked either, like do males that have actually lost many fights, do they become less effective at fighting, for example? Yeah.
We take one last question.
Yeah, I'm wondering why you go from like N to 2 to 3 to N to many. Is there some kind of phase transition in terms like mating behavior, or it's just simply continuous adaptation basically?
Yeah. No, I think it's a great question, and it goes back to this idea of like, it would be really fantastic to look at the complexity of of larger social networks. And I think I don't have anything really, we have not done it yet. And I think part of it's just because we're trying to still think about the granularity of behavior, like how much we can dissect, individual behavioral components versus thinking about social dynamics on a much larger scale. I think it's interesting that you do see actually very different behaviors emerge, even with just, with the addition of one. And I think, I would say anecdotally, when you add more flies, you actually do get like local networks, local interactions that I think will be quite informative and interesting. And in fact, there is evidence that even there's propagation of information across different, across a little population, for example, just by the kind of local interactions between flies. So if you give them, a choice between going to a good odor, an odor that they like and an odor that they don't, they're more likely to make the right decision if they're in a group because of these sort of local dynamics. But I think it's a very interesting direction to also pursue, yeah.